there were only 3 years where anybody associated with this project, the big project, 50 people working on it. There were only 3 years out of the 18 that anybody quantitatively measured root systems. That was Dan Walters. He's not here. Anyway, that's not atypical for water research. All right, so roots. Now let's talk about xylem and phloem. Xylem and phloem, vascular system in roots, in the stem, in the leaves, all over the place. The xylem vessels, the cells that make up the xylem, that transport in the xylem, are dead when they're mature. The cells that make up the transport system in the phloem are alive when they're mature. But in both cases, once you're inside this xylem vessel, or inside this sieve tube in the phloem, there are no membranes in that length. And so the movement of the fluid in the xylem and in the phloem is due to differences in pressure potential. The osmotic potential doesn't enter into it. It's the pressure potential that's the driving force for water movement, both in the xylem and in the phloem. So let's talk about xylem first. Responsible for water conduction. Okay. So, yeah, the cells that conduct the water have thick, lignified secondary cell walls, but no plasma membrane, no cell contents, nothing. They're just a cell wall. So, when the xylem transport cells are functional, they're dead, and everything goes away except the cell wall. They provide a low resistance conduit for water transport when they're dead. Xylem and phloem are always found close to each other in all vasculature. There are other cells in the xylem besides the conducting cells. So let me talk about 4 different kinds. The conducting cells are either tracheids or vessel members. And in addition to that, there are some cells that are fibers, and there are parenchymal cells, all affiliated with the xylem tissue. Tracheids are water-conducting cells that are found in gymnosperms and the so-called lower vascular plants. Right, gymnosperms, like pine tree, versus the angiosperm, like a sunflower. So gymnosperms only have tracheids for conducting water. They're hollow cells that are dead when they're mature. They have thick, highly lignified cell walls, and they have different shapes. They also have holes in the sides of them, called pits. Some of these holes are fairly simple, just an opening in the cell wall, like a space where the fibers are kind of far apart, so they make a hole there. Some of them are highly elaborated structures, but again, it's a hole. It's got some fibers and stuff going across the middle of it, but it's a hole. Here's a fairly elaborate pit. This is the front of Milburn's book. This is Abies grandis, so a fir tree. This is a pit in one of the tracheids in this fir tree. You can see this central region that's kind of thick, a bunch of fibers holding that torus to the rest of the cell wall, but no membrane or anything in between here. It's just an opening, so water easily goes through it. Just have a look at that. So tracheids are the only conducting cells found in gymnosperms and the so-called lower vascular plant. Now, these tracheids are lined up, typically lined up end to end. So here's one tracheid, and then here's another one that starts here, so they overlap. And the pits form the connections between adjacent tracheids. So these are single cells, but the next one may be right here that overlaps. And so what you wind up with is a fairly long conduit for transporting water. The other kind of conducting cells found in plants are called vessel members. And they're called this because vessel members or vessel elements are arranged end to end into a big thing called a vessel. The whole structure is called a vessel. They're similar to tracheids. They're lined up end to end, but the connections from one vessel member to the next vessel member aren't from these relatively small pits, but they're a great big opening between the two adjacent cells. It's a big hole. So that, I think I have some pictures. See it. So it's a vessel in the so-called higher vascular plants, in angiosperms and in ephedra, which is a gymnosperm that happens to have vessels, present even less resistance for water movement through such a structure than if you have the connections being a bunch of little holes between the adjacent. Typical dimension of vessel member, 8 to 500 microns in width, 200 to 2,000 microns in length. So, actually pretty big for a cell. They're arranged end to end to form vessels. The connections between the vessel members, the individual cells, are called perforation plates. And you'll see pictures of these. They're more or less big holes between the adjacent vessel members.
Question. So if I were to put this to a metaphor, it'd be like, one would be like a big tube or a ladder shaft, and the other one would be like segments of airlocks, almost?
Sort of, yeah, but it wouldn't have like the doors between the airlocks. Take
Okay.
the doors out, or
Sure.
maybe a screen door or something between the
Okay.
airlocks, the adjacent ones, yeah. Airlocks with screen doors, yeah.
Okay.
Versus a big shaft with a ladder in it.
Yeah.
Yeah, that's.
So it adds some kind of resistance, but not.
Right, not.
It keeps it from getting bad stuff in between.
Exactly right, exactly right. It keeps bad stuff from moving from one to the next. And by bad stuff, typically that's a bubble of air.
Yeah.
Yeah, an embolism. So xylem vessels and tracheids embolize quite frequently. And the plant has to make sure that embolism, just like in your circulatory system, that's bad news for you. It's bad news for the plant too. And it's in the plant's interest to keep that localized to where that bubble forms and not let it propagate all the way through the stem or something. And that's what those pits do. They're small enough so that they block an air-water interface from moving to the adjacent member. Okay, so in angiosperm, since we have these vessels that can be, the vessels can be a centimeter to 8 meters long. One vessel, 8 meters in this species of grape, right? This labrusca. The trade-off is that vessel has a fairly low hydraulic resistance. It's easy for the water to move all the way through that vessel because it has big holes, big connections between. But if it gets embolized, that whole vessel is cut off, not just the little trachea. Vessels are connected to each other end to end in overlapping regions where the connections are pits, these small little holes. So the pits are present in vessel members too. Here's a Fedra. Okay. Yeah. And plants that have vessel members and vessels also have tracheates. So it's kind of a redundant system. Okay. So no membranes, once we're in the vessel or in this string of tracheates, means that the driving force for water movement in the xylem are gradients in pressure potential. We can use this Hagen-Poiseuille equation as a first approximation for the hydraulic flux, the volumetric flux of water. The relationship between that and this pressure gradient. And remember, it has the R squared, has the viscosity on the bottom. So the bigger the vessel, the more flow you get for a given hydraulic, or for a given pressure potential. And again, it's a form of convective flow, laminar flow through large tubes, and for plants, that's what we have. We have laminar flow in the vessels and in the phloem as well. And the driving force is a difference in pressure potential. Okay. So here's some vessel members. Now you can see, these are the mangroves. I showed this earlier. But these are vessel members in different species of mangroves. So, for example, this one, big holes in the ends of the vessel member that connect to another vessel member here. So it's almost like having a continuous tube. Other ones show kind of holes shaped like this. So this is the perforation plate. This is the perforation plate for these vessel members. And these little black dots and little stripes here are the pits that are found in the side of the vessels and between adjacent vessels or between vessels and tracheates. All right, so let me get back here. All right. The pits themselves are small, 20 nanometers in diameter. That's really small. Usually that's small enough, as I say, to stop an air-water interface, which tends to separate the vascular system into little compartments, different components. And if one of them gets damaged, as you say before, like one of them gets a bubble of air in it, the pits are small enough so that that bubble of air can't move into the adjacent trachea or the adjacent vessel. It localizes that damage to one place.
How does bolus air happen? How does it happen?
These embolisms tend to form when the total water potential in the vessel gets very negative. And that total potential in the xylem is mostly the pressure potential. So these embolisms tend to form when the difference in pressure between outside the vessel and inside, or outside the trachea, outside the vessel, inside the vessel. That difference in pressure gets large enough, so for whatever reason, maybe there's a break, maybe there's a crack, maybe just one of these pits is really big, and air can that air-water interface can form, push its way in. They're very common. Embolisms are very common. I've got some graphs we'll see some data in a little bit, but they're very common. They're associated with low water potentials in the xylem, low pressure potentials in the xylem. They're very common in woody plants that grow in temperate climate, like here in Nebraska, when we go from summer to winter. So in the wintertime, a lot of the vessels in oak tree, for example, will be embolized and full of air. And then somehow, the plant has to push that air out and refill those vessels with water. That's the trick. The trick isn't getting the bubble in there in the first place. Hey, it forms just however it forms. The trick is now that bubble's in there, now what does the plant do about it? I'll say a few words about that. But yeah, I think it's just, it's associated with very low water potentials. So in dry soils, you get a lot more than in moist soils. And I think it's just because the vascular system isn't perfect. And the air can go in at any of these imperfections. Yeah. Let's just keep that. Okay. How fast does the stuff move in the xylem? About 1 millimeter per second. That's a typical rate of ascent of sap in the xylem. 3.6 meters per hour, about 1 millimeter per second. So, not real fast, but fast enough. Okay, so in woody plant species, so like an oak tree or something, the size of these vessels is different depending on the environmental conditions that prevail when they're formed. So typically you see big differences in the vessels that are formed early in the growing season versus late in the growing season. And so early wood, late wood, you see those names. You can see it in these cross sections, especially here in the oak, where you get a lot of small, a lot of big vessels formed early in the season, and then later in the summer, this part has small vessels, small diameter vessels. And this has been a way for classifying different kinds of trees. Here's another example. Here's 1 year growth in this red oak, I think. Northern red oak, yeah. So the vessels formed early in the summer are here, and late in the summer are here. And dramatic difference in the diameter, which plays into this equation we have, right? Because the plant's varying this R, we give it different characteristics based on what the gradients and pressure potentials are. All right, so conducting vessels. In addition to the vessel, the cells in the xylem that conduct water, there are fibers which mainly provide structural support to the vascular system. So here's a vessel here and phloem here and a bunch of fibers, right? So they stiffen up the stem. Thick lignified cell walls, dead at maturity, but they don't transport water. Fibers and parenchymal cells. So parenchymal cells are affiliated with the vascular system, with the xylem or the phloem. And these cells are not dead at maturity, but they, in a lot of cases, here is a parsnip root with xylem vessels, and these parenchymal cells almost completely surround all of the xylem vessels in this root. Their function, who knows? They may aid with lateral transport of substances into or out of the xylem. They also may retain their ability to differentiate into different cells depending on what happens. So phloem of the differentiation of some parenchymal cells following wounding of the phloem. And so, and who knows what the main function is, but they're always present in the vascular system always. There. So here's the quote of the day. Negative pressures, quote of the day, no, not in Milburn. Negative, these are the guys that, here's Milburn, the guy that wrote that book. And Zimmerman's one of the guys that made that pressure probe apparatus for pressure potentials in single cells. All right, they wrote some article somewhere that said, negative pressures can only survive in the xylem of plants for long periods of time because water is confined to small compartments. So, otherwise, plants wouldn't work. Cavitation, the formation of these embolisms, obviously is bad for the plant because if the vessel's full of water, if the vessel's full of air, the water can't get through there, right? So it decreases the hydraulic conductivity of the vascular system, reduces the hydraulic conductivity of the vascular system. And it's only because there's so much redundancy in that vascular system, a bunch of parallel pipes, some of which get embolized, but others don't. It's only because these embolisms are restricted to small compartments that the whole system is able to work. All right, these cavitation events occur at very negative total water potentials in the xylem, which is primarily the pressure potential component in the xylem. And you can get around them or circumvent their appearance by growing new vessels that are full of water, right? Okay, that one's okay. They can be refilled